So this is Genetic Technology Part 2, Chapter 15, Part 2, and it's Tools of Molecular Biology. So we'll be taking a look at electrophoresis, which we've looked at a little bit already, and polymerase chain reaction, which is what PCR stands for. So scientists want to make changes to DNA. They want to, right? In order to repair it or to make it better or things like that. So they look at what they know already. They know the structure of DNA, they know the chemical properties, and they study this to change DNA molecules. So they might use different techniques. They can extract the DNA from cells, they can cut the DNA into smaller pieces, and they now know how to sequence the bases in a DNA molecule. They also know how to make unlimited copies of the DNA in order to manipulate it and do experiments and study it. So in genetic engineering, biologists make changes in the DNA code of a living organism. Now remember, this right here is the genetic code I've got a picture of. And so they'll make changes in that in the actual organism. So one of the things they're able to do is extract DNA. And cells are open and the DNA is separated from the other cell parts. Then they cut the DNA using what are called restriction enzymes. Now remember, we have bacteria that can make these enzymes. And these restriction enzymes cut DNA at specific sequences. This drawing shows how restriction enzymes are used to edit the DNA. So here's that restriction enzyme ECOR1. Okay, and it finds the sequence GAATTC on the DNA, and it cuts in between the G and the A on both sides, and it produces what are called sticky ends. Remember that, okay? If you remember, rather. Then the fragments are placed at one end of the gel, of the electrophoresis gel, and the voltage is applied. And then when the power is turned on, the negatively charged molecules move toward the positive end of the gel. Okay? And obviously, the smaller ones are faster than the larger pieces. So this gel electrophoresis can be used to compare the genomes of different organisms or individuals. So it can also be used to locate and identify one particular gene in an individual's genome. So here is a picture of a DNA fingerprint. That's what the picture of. And you have a blood stain and samples of different people of their DNA. And we look to see whose DNA it matches. And it looks to me like it matches which one? That's right, John. Looks like it matches John. Here's the setup for the gel electrophoresis. So remember, we have the restriction enzymes cut the DNA, then they are poured into the wells on the gel, okay? And the voltage moves the DNA across the gel. Now, because the longer fragments of DNA move through the gel slow, they do not migrate as far across the gel as shorter fragments of DNA. So based on size, these DNA fragments make a pattern of bands on the gel. The bands then be compared with other samples. Okay? So here's where they get cut into fragments and put into the gel, the wells of the gel. Okay. The electric voltage is applied. And then, as you know, the smaller ones move faster and farther. And then what you do is you dye it, and then that's what darkens the bands. And those bands are used to compare with other DNA. Okay. Now, what do they do with this DNA sequence? They use it to study specific genes. They can compare them with genes on other organisms, and they try to figure out what the function is of the genes. One thing they can do with these sequences of DNA is they can cut and paste them. Now remember, we had what we call the sticky ends. So you can take that same section off of one piece of DNA and insert it into another piece of DNA, or DNA maybe from another organism or something like that. So these synthetic sequences can be joined to natural sequences. So you can say make that, and obviously they use enzymes to splice the DNA together. So enzymes also make it possible to take a gene from one organism and attach it to the DNA of another organism. And these molecules are called recombinant DNA. You've recombined different genes. And that's kind of how they got this. You may or may not remember this picture, but it's a human ear that was grown on the back of a mouse. And no, the mouse can't hear from it, but it looks kind of cool, I think. Now we're going to start talking about some stuff that we have not talked about before. And we have what's called the polymerase chain reaction. And this is how they make copies of genes. Okay? So what a biologist does is they add short pieces of DNA that are complementary to portions of the sequence. So if you have one strand of DNA, you get the complementary strand of it. So the DNA is heated, and that separates it into two strands. Then it's cooled, and that allows the primers to bind to the single-stranded DNA. And the primers kind of tell the DNA polymerase where to make copies or where to add those bases. Remember, DNA polymerase. That's what adds the correct bases. Remember that? Okay. So you have a DNA fragment that's to be copied. Now, if you notice, it's double-stranded right now. So they heat it to separate the strands. So the blue will be our original ones. Okay. Then the DNA polymerase adds the complementary strand. Those are the green ones. Okay. Then those two strands are separated. So you have the green on top and the blue on the bottom. And then the complementary ones, the new ones, are the red ones. Okay. So we've gone through the PCR once, twice, 3 times. And now we have 4 copies of that original DNA strand, don't we? So they can do it again. Split those. You have green and red, red and green, blue and red, and then red and green. And then those get split. Okay. Now we have 8 copies of that original blue strand. Okay, so here's the blue. You see the blue strand in the beginning. Step 2. Step 3, there's still some blue strands, right? And then step 4, there's still some blue strands. So this is called polymerase chain reaction, and is used to make multiple copies of genes. So if those get separated again, bam, we have 16 now, don't we? Okay, and this is the end of this section. So in class, we'll probably do some virtual labs having to do with this. So it should help reinforce this information.